Question 1. If a child has blood group O, it means that the parent genotypes are A, heterozygous A or B, B, homozygous A or B, C, heterozygous O, D, one of the parents is AB. Now, it's easy to knock out 2. There is no heterozygote O because O gene is recessive. So C is out. And if one of them is AB, there is no way the child will be O. So D is out. Now, A, heterozygote, either of them is heterozygote A, something like this, or heterozygous B. B, they are homozygous, so this one is out. So this one is out. The answer is A. Question 2. Human blood groups are determined by A, 4 antigens, B, 3 antigens, C, 2 antigens, and D, only 1 antigen. From our previous discussion, we know that we have 2 antigens, antigen A and B. That determines our blood group. So answer is C. Number 3. Which of the following can be the reason for carrying out test crossing? A, to determine the genotypes of the first filial generation. B, to determine the dominant allele. C, to determine the sex of a child. And D, to determine the genotypes of the parents. Now, to answer this question, you have to understand what is meant by test crossing. Test crossing is trying to breed an offspring of first filial generation with homozygous recessive parents. What do I mean by that? Look at a tall parent, homozygote tall, crossed with homozygote short. These people produce gamete for tall and for short. The first filial generation will have this and this combined and become this. And this is heterozygous tall, because if tall is dominant, the child will be tall. Now, you want to do back, you see the child short, tall, but you don't know whether the tall has the same gene with that of the father. So what you do is to test cross this offspring with homozygous recessive parents. This is the recessive parents. So this is the back crossing we're talking about. It now become the child's genotype crossed with the homozygous recessive parents. So if these people will produce short person, that means this individual has short gene. That is why a child with shortness can come from it. That is, if it is like this. If this one combined with this, it produces a short child. If this one combined with this, it produces a short child. So it means this person, it is not homozygous tall like the father or any of the parents that is tall, but heterozygous tall. The cross testing has proved it. So this is what the question is trying to find out from us. That what is the reason for test crossing? Therefore, the one that explains it is to determine the genotypes of the first filial generation. So A provides us with the answer. Number 4. Which of the following is sex-linked traits? A. Sickle cell anemia. B. Tongue rolling. C. Skin color. D. Color blindness. Answer is D. But what is a sex-linked trait? Is a trait, a character that is associated with X chromosome. X chromosome. And X chromosome and Y chromosome are sex chromosomes. For instance, this color blindness, you can illustrate it like this. This is the male crossed with a female. Color blindness, let's take H, capital H, for normal color and small h for the color blindness, because these traits are usually recessive. This child is normal. This one is a carrier. Female are usually carrier of sex-linked traits. Now, this Y is not having whether normal or the colorblind gene. This trait is not associated with Y chromosome. That is why, but it's associated with only X chromosome. X chromosome. That is why we call it sex-linked trait. So, in crossbreeding, this is H, this is O, this is H, this is colorblind gene. So if this one combined with this, we now produce a normal female, because XX is a female. If this one combined with this, you produce a carrier like the mother, H. If it is this to this, we have a normal boy. This O means nothing, and it's only Y that has nothing associated with it. So this is a boy. If it is this with this, we have a colorblind boy. So our answer is D, as said earlier. Number 5. The first filial generation of a cross between a tall and a dwarf plant was all tall. If the first filial generation was advanced to second filial generation, how many of 120 second filial plants would be dwarf? Very simple. Now, what to do is to have the tall plant cross with dwarf plant. They are all homozygous. Then they produce, this one produce tall gametes, this one produce short gametes, and the two produce heterozygous tall. That is what was all tall. All of them tall, all the offspring. Now, in second generation, you advance this one second generation. This is this cross with this. Then we have tall, short gametes, tall, short, or dwarf gamete. Then second generation, F2, will be this to this. We have heterozygous tall. This to this, we have heterozygous tall. This is homozygous tall. This is heterozygous tall. This to this, another heterozygous tall. This to this, we have dwarf. The question is, how many of the 120 second filial generation plants will be dwarf? If out of 4 you have 1, out of 4 you have 1, in 120, how many will have? Divide 120 by 4. That gives you the number, and that is 30. Answer is C. 30 of the 120 offspring.